Stem cells can generate themselves by self-renewal, and the stem cells are also capable to differentiate them into the mature cells. In many animal species, it is well known that the self-renewal and the differentiation of stem cells are locally regulated by a microenvironmental structure called niche. The idea of the niche was conceptually proposed by Dr. Ray Schofield in 1978. Then, the first example of the juxtacrine signal from the niche was identified by Dr. Alan Spratling in 1998, actually in the fruit fly Drosophila melanogaster. Nowadays, the niche concept was widely being accepted as a general and canonical concept to well explain self-renewal and differentiation of stem cells in many tissues of many organisms. On the other hand, particularly in the last decade, it has been revealed that self-renewal and differentiation are also significantly influenced by a physiological status of individuals, such as nutritional condition and hormonal balance. Our laboratory has been interested in the connection between individual physiology and stem cell behaviors at molecular, cellular, and systemic levels. During the course of our study using the fruit fly Drosophila, we have been getting interested in mating as an important external cue to influence stem cell behavior in female Drosophila. More precisely, what we found is that mating promotes female germline stem cell proliferation. This slide summarizes germline stem cells, or GSCs in short, in female Drosophila. Drosophila ovary is a spherical ball-like structure, as you can see here, and the tip of the ovary here containing the female germline stem cells. This is a magnified view. Female germline stem cells marked by these white circles are located at the most anterior part of the ovaries, which is called the germarium. These germariums are easily visualized by immunostaining with a specific antibody called 1B1 shown in green here, and then we can quickly count the number of the germline stem cells under a fluorescence microscope. In each germarium of the Drosophila melanogaster, there are usually 1, 2, or 3 germline stem cells. In this presentation, I'm using such a cumulative graphical representation indicating the ratio of the germarium population, which have 1, 2, or 3 germline stem cells respectively. In addition, the average number of germline stem cells is also represented as a number here, and the red bar here corresponding to the y-axis. What we found is that, as compared to the virgin female, mated female has much more germline stem cells by increasing the population of the germariums containing the 3 germline stem cells. In conjunction with other data that I skipped today, we conclude that mating promotes germline stem cell proliferation. You may think that the difference between 2.0 and 2.5 is very small. However, it must be noted that this difference could make the difference of the mature egg numbers such as 200 versus 250. Therefore, we think that mating-induced germline stem cell increase should be an adaptive post-mating response for reproductive success of female Drosophila. I'm showing this slide to summarize what we found until 2016. Mating-induced germline stem cell proliferation is mediated by a male seminal fluid protein called the sex peptide and its specific receptor called the sex peptide receptor. Sex peptide receptor is expressed in the neuron whose cell body is located in the female oviduct. In addition, sex peptide receptor neurons are necessary and sufficient to activate steroid hormone biosynthesis in the ovary. Then, the elevated ovarian steroid hormones are required for mating-induced germline stem cell proliferation. On the other hand, sex peptide receptor positive neuron, shown here, project into the ventral nerve cord, which is a part of the central nervous system, but not directly to the ovary. Therefore, there must be subconnections between central nervous system and the ovary to regulate germline stem cell increase. However, such connections were unclear 5 years ago. So the question to be addressed for us is what connects mating stimulus and the ovary to regulate mating-induced germline stem cell proliferation. In 2018, we published one paper demonstrating that meat-gut-derived neuropeptide F, or NPF in short, is essential for mating-induced germline stem cell proliferation. I skipped this topic today, but I just want to say here that this is the first report of the inter-organ communication from the gut to the ovary. In this study about meat-gut-derived NPF, we also found that NPF acts on the niche cells through the specific plasma membrane receptor for NPF, so-called NPF receptor. This finding raised a new question for us. So are there any other transmembrane receptor other than NPF receptor to regulate mating-induced germline stem cell proliferation? So to tackle this question, we conducted a transgenic RNA screen to knock down each of the transmembrane receptor gene by GAL4 driver active in the germline stem cell niche cells. We tested 288 transmembrane receptor genes that are encoded in the Drosophila genome. Then we counted germline stem cell number in virgin and mated females of each RNA condition. I need to make a long story short today, but among the receptor genes we tested, we focused on octopamine receptor, so-called OAMB, which is the 7-pass transmembrane receptor for the insect neurotransmitter octopamine. I'd like to remind you that octopamine is a counterpart of mammalian noradrenaline. So first, I'm going to show the phenotype of OAMB RNAi. So I'm going to, so in this experiment, we knock down the OAMB specifically in the germline stem cell niche cells by using the special GAL4 driver. In control animals, germline stem cells were increased after mating as compared to the virgin females, as I mentioned before. In contrast, knockdown of OAMB in germline stem cell niche cells impaired mating-induced germline stem cell proliferation. I skipped data today, but we also confirmed that OAMB RNAi resulted in the reduction of the phosphohistone H3 positive mitotic germline stem cells. So these data suggest that OAMB is required for the germline stem cell proliferation. So the next question is, where does the ligand octopamine come from? To answer this question, we focused on the 8 octopaminergic neurons that are previously described by Dr. Stefan Goodwin's group at Oxford in 2014. These neurons that Goodwin's group found is directly innervate to the ovary. The cell body of these neurons are located in the distal part of the abdominal ganglion, shown here, and this directly innervate to the ovary from the abdominal ganglion, as shown here or here in green. As I mentioned before, it is also known that the mating signal is received by a special subset of neurons called the sex peptide receptor positive neurons. Interestingly, the neurite of the sex peptide receptor positive neurons are very close to the cell bodies of these 8 octopaminergic neurons. Therefore, we examined whether sex peptide receptor positive neurons and 8 octopaminergic neurons are directly connected to each other? And the answer is yes, which was given by the GRASP assay. Briefly, this assay is based on the split GFP technology. If GFP is divided into the two small parts called the SPGFP11 and SPGFP110, either of them never exhibit fluorescence signals. However, if these parts are physically close, such as in distance of the neural synaptic connection, GFP is reconstituted and then GFP signal is recovered. When we express the membrane-tethered SPGFP11 in the dendritic region of the octopaminergic neurons and also membrane-tethered SPGFP110 in the axon of the sex peptide receptor positive neurons, GFP signal shown here was clearly observed in the caudal part of the abdominal ganglion, which correspond to the region shown here. These data suggest that the sex peptide receptor positive neurons seems to have synaptic contact with the 8 octopaminergic neurons, which directly innervates the ovaries. Based on this grasp of the observation, next we wondered if these 8 octopaminergic neurons are responsible for transmitting the mating input to the ovaries. To test this hypothesis, we manipulated neuronal activity only in these octopaminergic neurons using the special GAL4 driver transgenic lines and examine the germline stem cell numbers. When we activated these neurons using TRPA1 channel in the restrict temperature, germline stem cells was increased even in a virgin female, and the germline stem cell numbers were comparable to the germline stem cells in a controlled mated female. On the other hand, when we inactivated these neurons by using the tetanus toxin, germline stem cell increase was impaired even in a mated female, as shown here. These data indicate that 8 octopaminergic neurons projecting into the ovary control the germline stem cell increase in response to the mating stimulus. The next question is how octopamine signaling promotes germline stem cell proliferation. Previous studies have reported that the second messenger of OAMB is a calcium cation. Therefore, we assessed whether octopamine treatment triggered the calcium flux in the NIT cells. For this purpose, we conducted the calcium imaging using the calcium sensor probe GCAMP6. We dissected ovaries from virgin females and incubated them in the culture medium with or without commercially available octopamine. Then we checked the GCAMP signal intensity in the NIT cells in this ex vivo culture system. Please look at these videos. So RFP signal indicates the ovarian somatic cells, and the green signal indicates the GCAMP signals. In the case of the germline without octopamine, you can see the red signal, but there is no green signals. In contrast, when we added the octopamine in a culture medium, you appreciate the green signal which corresponds to the GCAMP signal, and this signal was frequently observed in the ovarian somatic cells. So this is a representative snapshot of the GCAMP images of the germline at the same time. You can appreciate again that GCAMP signal was detected in some ovarian somatic cells in the germline when we added octopamine in a culture medium. So this is a quantification. So octopamine treatment increased the GCAMP signal in the somatic NIT cells. In contrast, such increase in GCAMP signal was not observed in control treatment without octopamine. Moreover, importantly, the octopamine-induced G-CAMP signal was not observed in the ovary where OAMB was knocked down by RNAi as shown in blue. Taken together, these results suggest that octopamine treatment evokes calcium ion flux in the ovarian somatic cells through OAMB. We further confirmed the significance of octopamine and the downstream calcium flux for germline stem cell proliferation using this ex vivo culture system. As shown here, you can appreciate that the octopamine treatment for 12 hours in this ex vivo culture system was sufficient to induce the germline stem cell proliferation in ovaries dissected from the virgin females. The effect by octopamine is almost the same as what we observed in the mated females as shown here. In contrast, the ovary from OAMB RNAi virgin females did not respond to the octopamine treatment as shown here, suggesting that OAMB receptor is essential. Moreover, when we knock down the inositol triphosphate receptor INSP3R, which is involved in the release of the calcium ion into the cytosol, germline stem cell proliferation was not observed even with the octopamine treatment as you can see here. These results suggest that the significant role of intracellular calcium signaling in the NIT cells in octopamine-dependent germline stem cell proliferation. Even more persistent, we also took an experimental approach using the channel rhodopsin, which is photoactivated cation channel. We generated the fries overexpressing the channel rhodopsin called CS chromosome using the ovarian somatic NIT cell GAF4 driver, and then cultured the fry in the presence or absence of the all-trans retinal or ATR. As you know, ATR is required for rhodopsin protein function, so channel rhodopsin can be activated only in the presence of ATR, but not in the absence of ATR. When the dissected ovary was placed under orange light condition for 24 hours, germline stem cells was increased even in the virgin ovaries, as shown here. In addition, germline stem cell increase correlated with the increase of the number of the mitotic cells indicated by the phosphohistone HC positive cells, as shown here, as compared to the non-ATR situation. So these results suggest that optogenetic stimulation of the somatic NIT cells is sufficient for germline stem cell proliferation. And taken together with that I present so far, we that octopamine-dependent calcium signaling is both necessary and sufficient for germline stem cell proliferation. Our next question is how octopamine OAMB calcium access induces germline stem cell proliferation. In other words, we are curious of what is the downstream of calcium signaling to control germline stem cell proliferation. To answer this question, we focused on matrix metalproteinase 2, or MMP2 in short, as such downstream component. I'm going to explain the reason why in the next slide. Matrix metalproteinase 2, or MMP2, is a famous membrane-bound proteinase. It is well known that MMP2 cleaves a wide variety of extracellular proteins such as plasma membrane receptors and extracellular matrix. More importantly, Dr. Zhenzhen Sun and his colleagues at the University of Connecticut has demonstrated that octopamine and calcium-dependent activation of MMP2 is required for the follicle rupture process in Drosophila. We are impressed by Zhenzhen's observation and thus examined if this is also the case in octopamine-dependent germline stem cell increase. So to test this idea, we knock down the MMP2 using this GALFO driver, which is active, specifically active in the NIT cells. In the control animals, mating-induced germline stem cell increase was observed as shown here as compared to the virgin female. In contrast, the knockdown of the MMP2 in the NIT cells impaired mating-induced germline stem cell increase. So these results suggest that similar to octopamine-induced follicle rupture process, MMP2 is also required for mating-induced germline stem cell increase. We next conducted our ex vivo culture experiment to examine a relationship between the octopamine and MMP2. So here shows the control experiment, and octopamine treatment induced the increase in the germline stem cell number as compared to the mock treatment, which I already mentioned before. In contrast, MMP2 knockdown in the germline stem NIT cells impaired the octopamine-induced germline stem cell increase ex vivo as shown here. These results suggest that MMP2 act as a downstream component of the octopamine signaling to germline stem cell proliferation after mating. At last, we checked whether octopamine signaling enhanced BMP signaling because the GSC is maintained by receiving the BMP ligand from the NIT cells called the decapentaplegic in Drosophila. To estimate the BMP level in the germline stem cells, we measured the signal intensity of the phosphorylated MET, PMET, in the germline stem cells because this phosphorylation is essential for transmitting the BMP signal in the germline stem cells. Again, we took an ex vivo culture approach. This white line shows the outline of the germline stem cells, and we immunostain the PMAD using the specific anti-PMAD antibody. In the germline stem cells with the octopamine treatment, the PMAD level, shown in green, is significantly higher as compared to the germline stem cells without octopamine. Quantification of the PMAD intensity in multiple cells also demonstrate the increase of the PMAD signal by octopamine treatment as compared to the mock treatment. On the other hand, when we knock down the MMP2 in the NIT cells, the octopamine-dependent PMAD increase was not observed. And this is a quantification, and there is no statistical significance between the octopamine treatment and mock treatment in MMP2 RNAi condition. These data show that the octopamine signal is indeed required for enhancing the BMP signal in the germline stem cells. We also checked the PMAD levels in the ovaries that are optogenetically activated using the channel rhodopsin CM crimson. As you can see here, the optogenetic stimulation with all-trans-retinal and the orange light is sufficient to induce the PMAD elevation in shown in green as compared to the control situation. And this graph is a quantification of the PMAD intensity, and the elevation was statistically significant. Therefore, octopamine and the calcium signaling is both necessary and sufficient for PMAD elevation in germline stem cells. So taken together with all data that we got, we conclude that octopamine, calcium, MMP2 signaling regulates mating-induced germline stem cell proliferation. So when a female is mated with male, this 8-octopaminergic neuron projecting to the ovary are activated through the sex peptide receptor positive neurons. I skipped the data today, but we also have another result suggesting that these octopaminergic neurons express nicotinic acetylcholine receptors, which receive the acetylcholine from this sex peptide receptor neurons. And after mating, octopamine is released from this 8-octopaminergic neuron, and then octopamine is received by OAMB in the NIT cells. OAMB induces the calcium flux in the NIT cells, probably leading to the activation of MMP2, promotes the germline stem cell proliferation through enhancing the BMP signaling level in the germline stem cells. As far as we know, this is the first study showing that the germline stem cell proliferation is under the control of a direct neuronal input. Okay, so from this slide, I'd like to switch to the second part of my talk, which is about the relationship between the germline stem cell increase, and the dietary sugar. This project is still ongoing and quite far from complete. However, I would be happy if you could give me any comments and criticisms on this project. So this work is being mainly done by another talented graduate student in my laboratory, Ryo Hoshino. So from 2016 to 2020, our group has reported that there are 3 essential signals that regulate mating-induced germline stem cell increase in female Drosophila, namely ovarian equity steroids, mid-gut neuropeptide F, and the neuronal octopamine that I told you in the first part of my presentation. On the other hand, a number of previous studies have already revealed that mating and the germline stem cell maintenance are associated with the nutrients that Drosophila female flies consume. For example, mating accelerates the food consumption rate and also influence the food choice behavior of female Drosophila, which Carlos and Dr. Barry Dixon reported in 2010. In addition, Dr. Daniel Duramond Barbosa's laboratory at Johns Hopkins has published a series of great papers demonstrating that nutrients and the nutrient-related signals influence the long-lasting maintenance of female germline stem cells in Drosophila. Therefore, a natural question that we raised is whether any of the nutrients influence mating-induced germline stem cell increase. So first, we simply tested whether mating-induced germline stem cell increase is affected in low-carb, low-lipid diet condition as compared to the standard food condition. So the standard food that we used in this study contains water, 4% yeast extract, 0.55% agar, 9% cornmeal, and 10% glucose. In contrast, for making the low-carb, low-lipid diet, we simply depleted cornmeal and glucose from the standard food recipe, resulting in the lack of the major carbohydrate and lipid source. So we put the newly eclosed female flies of the white mutants, which actually used as a wild type in this study, on standard food or the low-carb, low-lipid diet for 1 day or up to 7 days. After this treatment, we let these female flies to mate as a males for 1 day, and they wing counted the germline stem cell number in virgin and mated females. These lines show the average number of the germline stem cells per germline 2 days to 7 days after eclosion on standard food condition. As you can see, germlines of the mated females shown in this dashed line have more germline stem cell numbers as compared to the virgin females as shown in the solid lines. This tendency is not changed over time, at least in 1 week after eclosion. In contrast, the situation was completely different when we used a low-carb, low-lipid diet. Mating-induced germline stem cell increase was still observed in younger flies, as shown here. However, mating-induced germline stem cell increase was impaired in animals that are reared in a low-carb and low-lipid diet condition for 5, 6, or 7 days. This cumulative graph represents the ratio and the average of the germline stem cells 6 days after eclosion, which corresponding to this region. So in the standard food condition, there are more germline stem cell number in the mated females as compared to the virgin females. However, in a low-carb, low-lipid diet condition, mating-induced germline stem cell increase is impaired. We also conducted anti-PMED immunostaining to examine the niche signal strength in the germline stem cell in the standard food condition and the low-carb, low-lipid diet condition. As you can see here, PMED intensity in the germline stem cells was elevated in the mated female as compared to the virgin female in the standard food condition. This graph represents a quantification, and the difference between virgin and the mated female are statistically significant. In contrast, PMED intensity was not elevated even after mating in the low-carb, low-lipid diet condition, as shown here. And this also supports the quantification analysis, and there is no significant difference of the PMED intensity between the virgin and mated female on the low-carb, low-lipid diet condition. These results support our expectation that nutrients significantly affect a mating-induced germline stem cell increase through the influence of the niche signals. Of course, the next question to be addressed was which nutrient is critical. Because of the time limitation, I'm just showing you our data suggesting that sugar should be critical. So in this experiment set, we first put a newly eclosed wild-type flies on the low-carb, low-lipid diet for 6 days. And after that, we placed these guys on plus glucose diet, which we just added the 10% glucose to the original low-carb, low-lipid diet. So the difference between this low-carb, low-lipid diet and the plus glucose diet is the presence of the 10% glucose. We then let these females to mate with males and count the germline stem cell number in virgin and mated females. Our results were actually really positive. And as you can see here on the right side, when we switched the low-carb, low-lipid diet from the plus glucose diet, mating-induced germline stem cell increase was restored by this sugar defeating. So these results suggest that sugar is critical for mating-induced germline stem cell increase. So from these results, we need to address how sugar influences mating-induced germline stem cell increase. A possibility that we easily expected was that the insulin signaling is involved in this process. And actually, we expected this should be quite likely as our original situation, our expectation. But I skipped the detail today. We have already examined whether the mating-induced germline stem cell increase was affected by a series of the insulin signaling knockdown animals, and we conclude that insulin signal is unlikely to be involved in this process. Another possibility is the glucose metabolic reprogramming in the ovary, which was very recently reported in the Nature Metabolism paper by Zeta and Carlos. This paper beautifully demonstrates that the glucose metabolism controls the sugar appetite as well as the mature egg production. We are so much impressed by this Zeta's paper, and we must examine if such glucose metabolism is also involved in the mating germline stem cell increase in the future. But so far, we don't have any data, and so, but I can just say that we must be examining that. We must examine this issue. But currently, in fact, we have been interested in the third possibility, which is the sweet sensation mechanism through the taste gustatory receptors, which I will tell you in the next slide. As you know, there are a variety of taste gustatory receptors in animal genomes. In the Drosophila melanogaster genome, there are about 60 gustatory receptors, and here 8 of them are known to be responsible for the sugar taste sensation, namely GL5A, ZL61A, and the 6 duplicated members of ZL64 subfamily. In 2014, Dr. Hubert Amrain's laboratory at Texas, USA generated the very interesting Drosophila mutants in which all of these 8 sugar receptors are deleted. This graph shows the neuronal activity in response to the several types of sugars in control shown in black and the mutants showing white. And you can appreciate that the activity of the sugar-sensitive gustatory receptor neurons are diminished in the mutant replicating all 8 sugar taste receptors shown in white. Based on this phenotype, the Amrain's laboratory named the mutant sugar-blind mutant. We obtained this sugar-blind mutant and counted germline stem cell number after mating. Interestingly, the results were really positive. So in control animals, which are the heterozygous of the sugar-blind mutants, mating-induced germline stem cell increase was observed. So I should say that this experiment was conducted in a normal standard food condition. However, sugar-blind homozygous animals shown here did not exhibit the mating-induced germline stem cell increase even after mating, as shown here. So these results strongly suggest that the sweet sensation by sugar taste receptors is required for the mating-induced germline stem cell increase. So shamelessly, that's all that I can share with you today regarding the second part of my project. I understand it is too early to say something bold from our current data, but our data so far imply that the sweet taste sensation through the sweet taste receptor somehow regulates mating-induced germline stem cell proliferation. We have no idea whether and how sweet taste receptor can influence the central nervous system, intestine, and or the ovary to regulate germline stem cell proliferation, but we optimistically believe that this research direction would open more complicated inter-organ communication to regulate the germline stem cell proliferation in the near future. So I'm almost closing my talk, and in this slide, I'm listing up the remaining questions that we are currently interested in. Of course, we are interested in which the question which substrate MMP2 cleaves to regulate mating-induced germline stem cell increase. Currently, we have no idea what is the actual substrate of the MMP2. And I don't show the data today, but we found that octopamine does not stimulate the ovarian egg-destroyed biosynthesis. When we found the octopamine as a trophic factor to regulate the germline stem cell increase, we originally assumed that octopamine may activate the ecdysone biosynthesis in the ovary, but this was not the case. So what stimulates the biosynthesis? There is a ton of the literature describing the trophic factor to activate the ecdysone biosynthesis in the prothoracic gland during larval development, but there's almost no information about the trophic factor of the ecdysone biosynthesis in the ovary. We are pretty much curious about it. And currently, we don't know how octopamine calcium signaling, NPF signaling, and ecdysone signal crosstalk with each other to regulate the germline stem cells. This is a quite important issue to understand the inter-organ communication at the molecular and the cellular levels. And we don't know whether and how sugar-sensing neuronal mechanisms involved in mating-induced germline stem cells. And as I said in my previous slide, this should open the new direction of our research. And we are also curious how these neurotransmitter and the hormones control the orientation of the germline stem cell division as a niche. Currently we just focus on the physiological events, but this cellular event is quite important to understand for the future studies. And we are also curious whether other neurons and hormones are involved in mating-induced germline stem cell increase. Many researchers have already described that the juvenile hormone, which is another important hormone to control the insect physiology, are responsible for some of the mating-dependent biological events, such as the enlargement of the midgut, which was published in Irene Miguel Arriaga's laboratory in the United Kingdom. So now some of the people are also curious of the possibility that the juvenile hormones are involved in the mating-induced germline stem cell increase and other reproductive process. And finally, we are of course interested in evolutionary conservation of the inter-organ communication mechanisms to regulate the stem cell proliferation and or reproduction beyond animal species. Although female germline stem cells do not exist in mammalians, including humans, ovulation process induced by the sympathetic nerves that produce norepinephrine or noradrenaline, and which is the counterpart of the octopamine, and also regulated by the female sex steroid hormone, which is the counterpart of the ecdysone. We believe it should be worth to examine the mammalian ovulation process is also affected, not only there's a sympathetic nerve and the steroid hormone, but also the unexpected neuroendocrine factors from other organs, such as the gut or the taste sensation organs. This should be the future and another future direction. Okay, so finally, so I'm closing my talk. So finally, I thank many people and the organization listed here for giving us a lot of materials. And I also thank these funding sources shown here to support our research activities. And lastly, I deeply thank all the past and current lab members in my laboratory, particularly Yuto, Ryo, and the former graduate student in my lab, Tomotsune Ameku, who actually started this whole project. Now he is a postdoc in Irene Miguel Arriaga's lab in the United Kingdom. So I stop there. Thank you for your attention, and I'd be happy to take any of your questions.
Thank you so much, Ryusuke. This was a great talk. Okay, so we have some questions here in our question box. So Azure Rose asked, thank you for being with us at now 2 a.m. This was very interesting. Is there an equivalent also for the parasympathetic system in the mammalian physiology?
Yeah, this is very interesting. This is a very interesting question, but currently we have no idea. The other neurons which correspond to the parasympathetic system can innervate into the ovary in the Drosophila. So that means that we don't know the negative regulatory system to regulate the germline stem cells after mating. So this is quite interesting. And actually, nowadays there are a lot of the database to describe the anatomy of the neurons, and recently there are very precise description of the neuronal connections, but this is just in case in the main central bodies, but not the peripheral part. So this is quite an interesting open question to describe the neurons innervating into the peripheral tissues. So yeah, this is very interesting question.
Okay. Thank you. Okay, so we have another question from Samuel Walker. He says, fantastic talk, really exciting results. The results with sugar-blind flies are enticing. Could you could the abolishment of the post-mating germline stem cell increase be due to reduced food intake in these sweet blind flies, or do you think it's the sweet perception itself, independent of intake, that is required for stem cell increase? And this was also one of my questions.
This is a very important question, and currently we don't have solid data to prove this point. We just do some easy cafe assay to measure the food intake, and so far we don't have any big difference of the food intake amount between the control animals and the sugar brine. But the cafe assay is not the perfect assay to measure the food consumption. So that is the reason why we recently introduced the fry pad from Portugal. So now some of the students are setting up the fry pad to confirm this point. So this is we are also interested in this point. And the second question is, so yes, so we don't have any solid data to discriminate the two possibilities, reduction of the food intake or adjusting. Acting in differently, acting directly on the germline stem cells.
Yeah. It's very cool. Okay, so we have a question from Carlos. We also have a comment from Carlos. Very nice and stimulating talk. Thank you for giving the talk at this late hour. And then he asks if there are any gustatory receptors expressed in the reproductive system.
And the short answer is yes. So we checked the single-cell RNA-seq database or other RNA-seq database describes the gene expression in the ovaries, and obviously some of the gustatory receptors, including the taste receptors, are expressed. So one possibility is that gustatory receptor in the ovaries are directly sense the sugar. But at the same time, as you know, the gustatory receptors are expressed actually quite wide variety of the tissues. And one interesting possibility is that the gustatory receptor in the intestine would have some function to control the germline stem cells. Particularly in the mammalian systems, some of the gustatory receptors are expressed in enteroendocrine cells, and that's responsible for controlling the secretion of the enteroendocrine hormones. This should also be the case in Drosophila or other organisms. So we are pretty much interested in the role of the gustatory receptor, not only in the neuronal system on the ovaries, but also in the intestines. We are currently tested this idea.
Okay, great. Okay, so we have another question from Juan Valencia. Thanks for this great presentation. I would just ask a curiosity, did you find any problem or issue regarding the effect that the white minus genetic background could have in terms of amino acid metabolism, mainly in terms of referees of journals of metabolism? Thanks a lot in advance.
Oh, yes. So I understand the question because the white encodes ABC transporter, and some of the research already described that the white ABC transporter is involved in incorporation of not only the pigment but also other components, including the amino acid or amino acid derivatives. But unfortunately, we don't have any solid data to prove this point, at least in our system. But what I can say is that almost all the data about the germline stem cell increase observed both the white background or white plus and white plus background, and there is no big difference, at least for our case. So we do not pretty much worried about the side effect of the white in our case. Okay. But of course,
so it shouldn't be a background. It shouldn't be specific for a specific genetic background, right?
Sure.
Okay, great. So we have another question from Carlos. I am very intrigued by your very nice data showing that octopamine directly affects germ stem cell number. This reminds me of the emerging work showing that in vertebrates, noradrenaline signal from neurons promotes tumor progression and metastasis formation. Do you think it's the ancestral function of the signaling system to promote neuron stem cell interactions?
Yeah, that is a very interesting possibility. I do believe that possibility as well. But of course, it's quite hard to compare the evolutionary conservation between Drosophila and mammals so far. As Carlos wrote, in the mammalian case, the people showed the connection between the noradrenaline and stem cells, but not the stem cells of the bone marrow and the hematopoietic system, but not the germline stem cells. So yeah, I do want to believe that the amino acid derivative neurotransmitter would have a very ancestral function to the stem cell proliferation, but of course, this is just an imagination, and we don't have any solid data. But that's quite intriguing for me.
Great. Okay, so we have a couple more questions, and we still have time. So we have one question more from Rita Figueiredo. She says, really interesting and clear talk, thanks, especially at 1 a.m. I might have missed it, but I'm wondering if a low-quality diet might lead to a decrease in female receptivity, thus changing sex peptide signaling and leading to the observed decrease in the number of germline stem cells.
Okay, thank you for asking for the important question. And I think that we can rule out this possibility because not all cases, but we sometimes check whether the female in a certain condition can really mate with the males by using the DJGFP strains. DJGFP strains is a very special Drosophila strain that can mark the sperms by the GFP signals. So they containing the green sperms. And when we mate the females with the DJGFP males, actually we can very easily visualize the green sperms in the abdomen of the females. And we checked if the DJGFP sperm can enter into the abdomen in standard food condition as well as the low diet condition. And we confirmed that after the 24 hours during the mating, almost all the female can mate based on the GFP signals. So we don't worry about the possibility that there's a kind of artifact of the non-mated status.
Okay, fantastic. Okay, so we have another question from Hyuye Sanaki. Thank you for a nice talk. About sugar-inducing stem cell proliferation, the flies are grown up in the food with sugar or without sugar, or food without sugar may reduce body size?
Okay, thanks. And so in our experimental condition, we grow the flies, we allow the fly to grow in a standard food condition just before the eclosion. So the difference of the food was made after the eclosion. So the bigness of the flies are of course the same, and the starting point of the diet change experiment. So maybe might be the reason why the mating-induced germline stem cell increase was still observed in the younger ages. This is just 2 days after the eclosion. We just observed the effect, more than 5 days after switch from the switch to the low carb, low diet condition. Maybe they accumulate some of the nutrients during the larval development, which I imagine.
Okay, fantastic. Thank you so much, Ryusuke. It was a great talk. It was a great beginning of the year for our brain-body interaction series. So if someone else wants to join us and continue to discuss with Ryusuke, please just drop us an email into our Brain Body Interactions email, and I will send you the Zoom link. Meanwhile, thank you all for being here with us today, and I see you next time. Ryusuke,
thank you very much.
We see you in a couple of minutes in Zoom.
Sure.
Thank you.